Okay, so welcome back to part 4 of chapter 22. This case, we're talking about clonal selection. We were talking about how the cells, the T cells, B cells are selected. Once they're selected, then they get cloned. In other words, that cell has been determined to be special because it could detect something that was a foreign particle, something that we want to create a immune response to. And so it's selected for clonally. It is cloned. It is proliferated. That one cell gets proliferated um, until there are lots and lots and lots of them. Okay. The first encounter between the antigen and the lymphocyte uh, usually occurs in the secondary lymphatic structures, as says there, antigen and blood taken to spleen. Um, so the antigen comes in, and the first response that it gives, there we go. First signals direct contact with the MHC molecule of the antigen presenting cell. So we're talking MHC class 2 here. If it doesn't recognize the antigen, T cell disengages quickly. If it does, it binds to it. Okay. And this binding stimulates the T cell to proliferate. Okay. Um, and other receptors, the APC and T cell interact. Helper T cell secretes interleukin 2s to stimulate itself to proliferate. Um, and it also stimulates other T cells. So proliferation, forming clones of helper T cells with the same T cell receptor. So here's that in a graphical form here. So this is what we're talking about here. First signal CD4 binds. So the antigen and the antigen presenting cell causes it to release IL-2, interleukin 2, and that induces itself. So it's inducing itself after it's gotten activated by binding to the MHC class 2 molecule. Okay, an antigen on an MHC class 2 molecule, not just the molecule itself, but binds to an antigen that gets presented to it on that. All right. So again, there's the first signal direct contact um, for cytotoxic T cells. Direct contact with T cell receptors, cytotoxic T cells on the MH1 molecule. Interacts stabilized by CD8. So if it's a cytotoxic T lymphocyte, it will recognize the antigen there, of course, on the CD8 signal. So their IL-2 that came from the helper T cells binds the cytotoxic T lymphocytes and causes them to proliferate. So you get a T cell response. So you're getting it in the helper T cells and the cytotoxic T cells. So the proliferation occurs, and you get many of them to go out there and fight the disease, fight the infection. Now, some of these will actually differentiate some more and become activated, like it says, cytotoxic, become memory cytotoxic T cells. So it's the memory cells that stick around afterwards, and it's when we give ourselves immunizations, when we get immunized against something, that's what we're basically creating is memory T cells and memory B cells that will then hide out. They will migrate to and wait in the secondary lymphatic tissues, lymph nodules, and then the lymph nodes and the spleens and the tonsils and all that. They're waiting to encounter it again. So when they encounter it again, they'll be able to launch a cytotoxic T cell response and make antibodies. Okay, um, and the antibodies, will fight the disease. So, but they come on very quickly. So we'll talk about that a little bit later. So here's the cytotoxic T lymphocytes. First signal, CD8 binds with class 1 infected cell, interacts with antigen within the class 1 MHC molecule. Okay. Second signal, IL-2 released from activated helper T cells activates cytotoxic T lymphocyte. Okay. So, and then it proliferates. Activation of B lymphocytes, kind of similar. Intact antigen binds to the B cell receptor, which cross-links to B cell receptors. Stimulated B cell engulfs process and presents antigen to helper T cell for recognition. So, a stimulated B cell, they before they differentiate to become cells that we call plasma cells or cells that make antibody, they will engulf. They're phagocytic cells and they engulf things and they present those particles. Okay, just like the other antigen presenting cells, they present them to the T cells. Okay, to try to activate them. And when they find one, they do activate them, and they activate the B cell as well. So you can see here the B lymphocyte, okay, has picked it up, activated the T lymphocyte. Okay, there's MAC2, picked it up on the B lymphocyte. IL-4 then, interleukin-4, a different molecule, will act upon the B cell and cause it to proliferate. Okay. In proliferation, some of them become memory B cells, as you can see, and some of them become plasma B cells that make antibodies. So remember over here, it had to phagocytize the thing. It doesn't do that once it gets over here. Once it's a plasma cell, it won't phagocytize anymore. It's differentiated, okay? But initially, it has to get the response, to get the stimulation to cause the response. So most differentiating plasma cells and memory cells. Okay. After a period of time, the lymphocyte exits secondary lymphatic structure, circulates through blood and lymphadase, makes it more likely lymphocyte will encounter specific antigen. All right. Okay, so now we're looking at what we're calling the effector response at the infection site. So now that we've kind of defined and we've been looking at all the components of the immune system and how they get all activated, we kind of look now kind of back to the secondary along with the innate and the adaptive immune systems, and we kind of combine them all together and look at what happens to cause an infectious disease and what causes our ability to fight them and win, basically. Mechanisms by lymphocyte to eliminate antigen. So each lymphocyte has also what do the helper T cells do? Release IL-2, IL-4, cytokines. Cytotoxic T cells, they destroy unhealthy cells by apoptosis. Plasma cells produce antibodies. As we've already said, effector response help activate B lymphocytes. So we're talking about the T lymphocytes again. Activated memory helper T cells migrate to the infection site. So the cytotoxic T cells, they're going to target the infected cell themselves or the infectious agent directly. So they've got to get to the site of infection. They've got to get over there. So they leave their cozy little spot there in the lymph nodules and secondary lymphatic tissues. Find the site. Okay. Cytotoxic cells of effector response activated. Memory cytotoxic T cells migrate to the infection site, destroy affected cells, display the antigen. Because it works against antigens associated with cells called cell-mediated immunity. So this is cell-mediated immunity. It's a cytotoxic T cell. It goes after cells. All right, so that we call that cell-mediated immunity. And what it does is it will blow holes in it and it will induce it to kill itself. So apoptosis. So looking at the B lymphocytes, most of the B lymphocytes that become active that are activated will become plasma cells, all right, and create antibodies. Let's take a minute and look at antibodies. What is an antibody? An immunoglobulin, as we call them, Ig. And it does several things. It causes the inactivation of the target, so it can take the target, which is the antigen, and bind to it and inactivate it. If this antigen is something on the surface of a cell, of a bacterium, of a fungi, something else like that, then it can target that specific organism to be attacked by the immune system. Okay, so it can do several things. It can do both of those things. So let's look at the shape of it. Okay, so I've got this Y shape here. Okay, uh, heavy chains are the big ones, and light chains are the little ones. So the heavy chain is part of these parts, the arm regions. Okay, the heavy chain, light chain are totally within the arm region, but the two heavy chains continue on to the constant region, like the stalk of the Y rather than the two arms of the Y. Okay, the stalk is the constant region, stem region. So why is that important? Because that will recognize, that will target. It's like a flag, okay? It's basically flagging something and calling in the immune system to come and attack this thing that it's bound to. Okay, so it looks for the flag that's that's planted in the tissue to go over that flag. And if the flag is not, whatever flag it's expecting it to be, they're going to ignore it. They're not going to go over there. So those are the two regions. So what can antibodies do? They can neutralize, like I said, they can cover the antigen. They can cover that part of the organism that gives the antigen and stop it from causing infection. They can clump, they can agglutinate, they can make this massive complex of um, antibody and antigen and organism, foreign organism, that they basically take it out of commission. Okay, um, they can precipitate, so something that's not soluble anymore. They can make that found by phagocytes. Um, so there's those three: neutralization, agglutination, and precipitation. They can help complement. A minute ago, not a minute, but a couple of presentations ago, mentioned complement. Complement is one of those proteins that is just kind of has variable types, and they just go through the blood. They're always in the blood. They're in the plasma of the blood, and when activated, they will kind of leap to action and do the things that the complement does. Remember, we talked about some of those things. Help to target something. Uh, might make a hole in the membrane. All right, opsonization. Some phagocytes have receptors for these. The FC region antibodies makes it more likely to target cell where to be seen. So opsonization, again, that's the flag action. They can activate or trigger natural killer cells. All right. So there's just those things there. Yeah. So here we go with the different immunoglobulins. There are 5 different classes of immunoglobulins. Most of them are the IgG, so IgG, IgM, IgA, IgD, and IgE. Most of the antibodies are IgG. IgM, it's a pentamer structure. Oh boy, don't have a picture of it. IgA found in areas exposed to environment produced in saliva, mucus, tears, breast milk. Um, it's a dimer, so it's two Y regions whose two Y antibodies whose constant regions are bound together. Okay, so either end of the molecule has got the arms of the Y structure. IgD, antigen receptor for B cells. IgE, um, usually used against parasites. Okay, otherwise not much. It attracts. It says there as it says, it attracts eosinophils. Remember eosinophils? Um, they can cause irritation in different parts of the body. I heard of eosinophilic heartburn, and and there are other diseases too that are eosinophilic. So they rely or they're something that's up with the eosinophils being overactive. Okay, so they can switch from one class to another. The antibodies can. Now, mention memory cells. Talk about those a little bit. A memory cell is just a cell that it basically goes into hiding, goes into lymphatic secondary structures, and it just waits for the next encounter. So there's a time lag between the first exposure and direct contact. Adaptive immunity requires contact between lymphocyte and antigen. You need the antigen presenting cells to kind of get in on that. It takes a little while, so it takes a couple of weeks. So the first time you're exposed to something, you will catch the disease, okay? Unless you're immunized against it, because the immunization is meant to mimic that first time. And the first time is when you start to make antibodies. And so then the second time those antibodies get made, boom, right away. And the T cells go into action, boom, right away, before the disease even has a chance to take hold and cause disease, before the infectious agent even has a chance. So you get a powerful secondary response. That's a secondary response. So the second time, every subsequent time, not just the second, but every subsequent time. The first time it takes a while. That whole process that we saw has to unfold. The selection, negative selection, positive selection, interaction with the MHC class 2, all that has to happen before the cells will start to proliferate, and then those cells can go out and fight the disease, and it'll go away. I mean, everyone hopes. But the memory cells go off, like I say, they go off into the secondary lymphatic tissues. They wait for the next time. And then the next time, they don't have to go through all that rigmarole, as they say, of MHC class 2 being activated. They find the antibody right away and they activate. That's it. The antigen, sorry. They find the antigen, meaning that the body's been exposed to this again. They just leap into action, start making the antibodies. The B cells do start proliferating. The C cells, the C cells, cytotoxic T cells will. All right, so here you can see the intense difference between them. The primary response, the first time, you don't get a very big response here. Second time, you get a huge response, and you get it a lot quicker. Okay. How to measure something called a titer. We look for a place like a distance migration pattern through gel, and when we see that the antibodies have migrated enough to cause a big complex of molecules to coagulate or agglutinate, then we see it in the plate. Okay. So one of the things we talk about the innate immunity and the adaptive immunity. Remember, innate had two parts. First was the barriers, just the barriers, mucous membranes, skin, whatever the barrier was. The second one was the part of the innate, and these are basically like the granulocytes, uh, the neutrophils, basophils, and eosinophils. Okay, they work because there is some kind of an infection that has occurred, some kind of breach, some kind of infection. They don't know what it is. Okay, but then the third line, which is the adaptive, then that's the lymphocytes. They know exactly what it is they're looking for and exactly what it is they've encountered, and they're ready for it, and they go out. Okay. Um, but we can also split it up in terms of active immunity and passive immunity. So active immunity occurs when an individual has a direct encounter with the antigen, with the protein, and the whole thing that I just described all unfolds. There's also passive immunity, where you get that from another individual. You can get the antibodies from another person, okay, that will help fight it. This is what they're doing with COVID now, actually. They used to do this a long time ago, like in the turn of the last century, 100 years, 100 plus years ago. What it was people didn't move around very much, so they like it would happen in valleys. The sickness would kind of sweep through one town and then the next and then the next kind of. So one town that had an epidemic, an outbreak of it, okay, they try to quarantine people so that it didn't go somewhere else, but inevitably it would. So in the next town where it broke out, okay, in order to treat the people who had that, they'd go to the previous town or a previous town to people who had just gotten over it, and they'd extract their blood plasma. And in doing so, they were collecting their immunoglobulins, their antibodies to this um, organism, to this disease, and they'd administer them to somebody in their town that now has the disease. Okay, this is exactly what they're doing with COVID, because they wanted to know people who have it. If people who were tested positive, they're saying, well, you're making antibody, and especially if they weren't symptomatic, they would take their blood and get the plasma from it and get the antibody from it and give it to somebody else who was suffering with COVID, and they had some success with it, and why shouldn't they? This is an old method. We kind of fell out of sorts with it. Kind of didn't haven't done it because there really wasn't reason to, and mostly because too people would get diseases. They could get. You never know what they were going to catch along the way. Take plasma from one person and give it to another, inject it into them. That's kind of how a lot of people caught AIDS. Basically, giving blood. People who had the HIV virus would give blood, and that blood would be given to someone else, and they catch it. So similar kind of thing there. They don't know, they can't tell what might be there, and so it's best not to do that. But again, the consequences if you don't are likely that someone's going to die. So I guess that's better than contracting another bad disease. So you know deal with that then is the next step if it happens, I guess. Um, and that's a passive immunity. The person gets an immunity, gets antibody, and is able to fight the disease, but that person did not make those antibodies. They got them from somewhere else. Another way that can happen is infants through breast milk can catch them, can get the antibodies also. So there's a rundown between active and passive immunity. Again, this is what vaccinations are all about. This is what immunizations are all about. They are giving us, when we get an immunization, an inactivated form of the virus. In the old days, they take the virus and they grow it up, usually in a eggshell that was fertilized, because the cells would be growing in there, right? It's fertilized. It's a nice little, it's its own little ecosystem, its own little contained system in there. So you just inject it in there, so the cells that were growing would take the virus up and grow the virus. So you had like a lot of virus, okay, in the end. And then they take the virus that they were collecting, they deactivate it. They could heat kill it. They could treat it with a chemical, but they couldn't destroy it too much. They had to destroy it enough so it wouldn't cause disease anymore. But if they destroyed it too much, then the person who would be getting it wouldn't have an immunological response to it. It's too destroyed and it can't find the antigen. So it was a fine line, and as far as I know, there's still some companies that make it that way. They might not sell in the U.S. or the Western world anymore, but the COVID ones were all molecular. So the COVID ones were all basically produced in the lab. Okay. Um, so anyway, and that's what we're doing when we get immunizations. The doctors, the healthcare, the shot is inducing us to simulate the disease because we're getting exposed to the antigen. And then the whole thing happens, and you get the memory T and B cells. So that now you're chock full of memory T and B cells, so you're not going to catch the disease, at least in theory. Nowadays we're getting viruses that these things don't, that they work on, but the virus has means of mutating itself so that it can stay a little bit ahead, which is really a bummer because it's a great way of you know fighting the disease. And then we got what we call hypersensitivities, basically allergies. Okay, um, so allergies, things that are foreign, but they're not disease causing. Our body mistakes those for things that it's supposed to launch an immune response against, so we get allergic to them. So acute hypersensitivity and allergy, acquired immunodeficiency, AIDS. Okay. Well, all right. So there we are with the last part of this chapter. It's kind of a long chapter, very fascinating. Go through it, read through it a lot, understand it. Definitely ask questions because it is a little confusing. It's a big deal, um, but it's an important one. So good luck. Bye.